So activity 5 is the alveolar model. Note that this model is enlarged so you can see the alveolar sacs. The central tube is not the trachea, but rather and this should say less than 1 millimeter wide, terminal bronchiole branching to form respiratory bronchioles. So this model is also known as the microscopic lung model because it has been enlarged so you can see the individual alveolar sacs. Otherwise, you would not be able to see them this clearly and in this much detail with your naked eye, because these are microscopic structures. So the first part of the activity says to locate a terminal bronchiole with its supportive bluish cartilage. When the terminal bronchiole bifurcates, we get respiratory bronchioles. Note the circular bands of smooth muscle, which are pinkish on the smaller respiratory bronchioles. Without supportive cartilage, these tubes can adjust airflow to the alveoli. So you can see that here. This first one right here is going to be a terminal bronchioles. Terminal bronchioles come first in our airflow for the respiratory system. So the terminal bronchiole here, these would be respiratory bronchioles. And then once we get into the alveolar sac here, this bunch of alveoli, it is called the alveolar duct. Now it says to notice the blue supportive cartilage here. But once we get down here to the respiratory bronchioles and the alveolar ducts, you'll notice that there isn't any more of this cartilage. Your larger bronchioles and your trachea, bronchi and trachea, they're all going to require supportive cartilage to keep their structure. However, as we get farther down the respiratory zone, the respiratory bronchioles and the alveolar ducts will not have that cartilage. They'll only rely on smooth muscle because this allows them to adjust their size in order to regulate airflow. So blue cartilage up here, but not so much down here. Actually, not at all down there. And so the next thing says, name the medical condition where these bronchial tubes sometimes narrow upon exposure to a respiratory irritant. So you can imagine that if these become irritated, they're going to also become inflamed. And so the inflammation of bronchial tubes, we would call that bronchitis. And so we'll go through the learn list here. We already showed the terminal bronchiole, which comes first with the blue cartilage, then the respiratory bronchiole, which is mostly smooth muscle, and then alveolar duct, alveolar sacs, and alveoli. And singular would be an alveolus. So again, we'll go through it again. Terminal bronchiole here, these are respiratory bronchioles. Once it gets into the sac, and each one of these groupings of alveoli are alveolar sacs. Once we get down here into the sac, it's called the alveolar duct. You can see a little bit here as well. And then these would be individual alveoli. Singular would be an alveolus. And so the next part of the lab says the specific tissue type is simple squamous epithelium. Cells release surfactant, and you should know from lecture that it is type 2 alveolar cells that release surfactant to decrease hydrogen bonds. By decreasing hydrogen bonds, you lower the surface tension of water, and you make it so that the water molecules are not as attracted to each other, so it keeps those air sacs, those alveoli, nice and open. What is the PO2 of air in here? PO2 should be 104 inside the alveoli. PCO2 should be 40. So 104 millimeters of mercury and 40 millimeters of mercury. Next is the stroma. It makes elastic fibers, which are actually only going to be the black on this model. And it's for compliance and stretchiness of the lungs. And systemic capillaries deliver oxygen and nutrients to the stroma. So the stroma is the supportive network of an organ. So like we talked about the lymphatic system, the stroma for that system is going to make reticular fibers that act as an anchor for cells trying to carry out immune functions. The stroma for the lungs is going to be a little bit different because the lungs function differently. So we have, this is going to be elastic fibers here, some connective tissue fibers for structure, and then capillaries there. So that would make up the stroma for the lungs, and the elastic fibers are really what's going to contribute to the lungs' compliance or stretchiness. The more compliance the lungs have, the easier they are to expand, the stretchier they are. And the next part is going to be the visceral pleura. Specific tissue type is simple squamous epithelium, and it produces serous fluid, as does the parietal pleura seen on the torso model. And so the visceral and parietal pleura make up the serous membrane of the lungs. And so that's going to be these cells out here. These cells are going to represent the visceral pleura, which is going to produce serous fluid, which is going to reduce the friction between the visceral and parietal layers, so that when the lung expands and those two membrane layers rub against each other, they don't cause friction. So slide or glide against each other with the serous fluid. Okay, next is the pulmonary arteriole. Think, what color is this and why? The pulmonary arteriole actually is a small branch that comes from one of the pulmonary arteries, which came from the pulmonary trunk, which came from the heart. And so the pulmonary arteries are going to be taking the blood to the lungs where gas exchange will occur in order to reoxygenate the blood. So these pulmonary arterioles should be blue because they are deoxygenated. They are the ones that are on their way to the lungs for that oxygen. And so this blue vessel here is a pulmonary arteriole. Again, it's a small branch off of one of the pulmonary arteries. And so these will eventually lead to the capillary bed, where they're going to form the arterial side of the capillary bed for gas exchange to occur. And then it asks the partial pressure. So the PO2 of deoxygenated blood in this blue vessel should be 40 millimeters of mercury, while the PCO2 should be 45 millimeters of mercury. And next are the pulmonary capillaries. Color changes blue to red as red blood cells become oxygenated. So blue arterial side here, and then we get to the capillary bed where they're going to be red because now they're oxygenated, and the red vessels will make their way up. And so the next one says pulmonary venules. Think, what color is this and why? Pulmonary venules will eventually become the pulmonary veins, which are taking the now oxygenated blood back to the heart. And so it should be red because it is oxygenated. So this right here would be a pulmonary venule, which will eventually form the pulmonary veins to return this now oxygenated blood back to the heart. And so the PO2 should be 100, while the PCO2 should be 40. And I believe that is all for activity 5.